ATP. Is ATP a nucleotide? Yes or no? I have a head shaking yes. Yeah, right? It has all the parts that define nucleotide. It has adenine, a nitrogen containing base, attached to a 5-carbon ribose sugar, and then it has 3 phosphates on it. So technically ATP is a nucleotide, but we don't think about its role in, sort of polymerizing into DNA or RNA. Is how we think of ATP. Okay. So important thing here, this figure is sort of showing us we have ATP. It's showing we're forming ADP, adenosine diphosphate, and a phosphate ion popping off. So this bond right here being cleaved. And it's saying that what's coming out is energy that is available to do some type of cellular work, right? So we said maybe that's pushing concentration gradient, something against the concentration gradient, pushing ions against their electric gradient, or for synthesizing something else that's not favorable. And to put it back together, we need an input of an energy source from like food or sunlight. How does this make sense given that I'm breaking a bond and I'm telling you energy comes out when I spent most of last time trying to convince you that that doesn't happen? Thoughts? Thoughts on what's missing from this figure to explain what else is going on? Yes?
An enzyme.
An enzyme. Great. What else? What other things aren't being considered? There's some other reactants, some other things that are part of a bigger reaction than just an isolated breaking of ATP all by itself, right? Moreover, that energy that comes out, and maybe you guys have it memorized, maybe you don't, it's like negative 30-ish, 31 kilojoules per mole is what you get when you break down a mole of ATP. That is delta G. It is Gibbs free energy. And so what we were always talking about was exclusively enthalpy. So we weren't there yet. There's no consideration for entropy going in when I say we just cleave that. And that delta G term equal to, actually here it is, negative 30.5 kilojoules per mole. It has, and we'll talk about this notation later. Ooh, the prime and the not, so fun, love notation. It's fun. Yes, we haven't talked about entropy, we haven't talked about temp, and we haven't talked about the fact that most of this stuff, first of all, that picture was oversimplifying. We didn't think about the fact that this happens through hydrolysis, so a water molecule is involved. Okay, so it's often drawn with like all of that extra off of it. It's just ATP to ADP and then energy comes out. A little misleading. Even here, then it's showing how you can go down again to a monophosphate. You could have, a pyrophosphate, this PPI ion hanging out. Even this drawing here is not accurate. H plus doesn't really hang out like this. It tends to want to be hydronium and be H3O with water, so new interactions are forming. So really, it's just an oversimplification where we haven't considered all the new interactions that happen once we pull them apart, okay? And also at a very more just basic level, entropy went up here. I went from 1 thing to 2. True story. So that delta S term going up also helped contribute to delta G, and also all the water sort of shielding and encasing this stuff is involved, as well as whatever is happening, okay, on an enzyme such as a protein kinase. Protein kinase can bind a protein, it can bind ATP, and it can move one of these phosphates. And again, we cover this in more detail in chapter 12. So you don't have to memorize what a protein kinase is right now. But again, it's important to remember that this enzyme binds its protein, it binds ATP, it moves the phosphate over, and then things are dissociating, and overall that's very favorable for it to happen. Okay? But just breaking one bond, misleading.